Doesn't that just make you feel like you're happy to be alive, you know? It's kind of like taking an exam, which is coming up on Tuesday, right? If you're not alive, you can't take the exam, so, right? Well, you can. I've had people that probably, never mind, that's not matter. Anyway, so we have an exam coming up on Tuesday. I've had a couple questions. One, do I post old exams? I do post an old exam, and I meant to do that already, and I haven't done it. So I post the old exam to give you information on the format of the exam. And that's why I post it, all right? You will find that I don't post other old exams. And the reason I don't do that is I find students study old exams. And that's a really dumb idea because, as I tell students, exams change, material doesn't change. So what happens when I post an old exam, if I do something and then let's say I change a question, and the student has memorized that question, and then they see that it's different because they weren't paying attention, their impression is always the following: you're being tricky, right? Well, I don't ask questions to be tricky. I, honest to God, don't do that, and I hope that you guys know that. So I don't post old exams in general for that purpose. I post old exam, and I will post one old exam for this exam. I won't post it for the other ones because the format will be the same on all of them. But I post it so you know the format, okay? So that's the deal, and I find that works pretty well. I know there's old exams floating around out there and so on and so forth. I hope that you're not studying for exams by studying old ones because that's not a good idea, okay? I will do a review session also. I haven't looked at my calendar yet, but I will announce that on Friday. I don't usually like to do it the night before the exam, which would be Monday night. So I might do something on the weekend. I haven't decided, but I'll take a look at my schedule and so forth and do that. Yes, I do videotape the review session, so if you can't make it and you want to watch that, that's fine too. Okay? All right. We have some material to get through. So let us dive right into the material. The first thing I want to talk about, I finished talking about the tryptophan operon yesterday, and you guys were looking pretty dead by the time I finished it, so I felt sorry for you and didn't go into the next thing. So today I'm going to go into the next thing. All right? So let me say a little bit about eukaryotes, and I'm going to do eukaryotes kind of like I did with respect to DNA replication, and that is I'm going to tell you some of the differences, but certainly not all the differences, because there are many, many differences regarding transcription in eukaryotic cells compared to prokaryotic cells. Prokaryotic cells are very simple, and they're simple because there's naked DNA floating around in basically a cytoplasm. In eukaryotic cells, we've got histones, and we've got DNA wrapped, and we've got coils, and we've got chromosomes, and there's and we've got more genes. And we've got a lot more DNA, and there's a tremendous amount more complexity. So I just want to give you just sort of a scratching at that surface of complexity, and then we're going to move on, okay? And we'll talk about translation. All right. Now, with respect to transcription, we start out with some very basic differences. We talked about there being one RNA polymerase in prokaryotic cells. There are at least 3 in eukaryotic cells, okay? There are at least 3 in eukaryotic cells. They're called RNA Polymerase I, RNA Polymerase II, RNA Polymerase III. The one that we will focus the most on is RNA Polymerase II, because RNA Polymerase II is what makes the messenger RNAs. The RNA polymerases are broken down by function. Okay? So RNA Polymerase II makes messenger RNAs. RNA Polymerase I makes ribosomal RNAs, and RNA Polymerase III makes transfer and other small RNAs. Okay, so we see a breakdown by function in terms of the RNA polymerases. RNA Polymerase II, which, as I said, is the one that makes the messenger RNAs, is fairly complex. This is, excuse me, this is a listing of the individual protein subunits of RNA Polymerase II. There's a lot of them, okay? We don't need to worry about a lot of these. I'm going to talk about one of them that isn't even listed on here, in just a little bit because it works with RNA polymerase II to help it recognize promoter sequences. Remember, we had a sigma factor in the prokaryotic cells that did that. There is a protein in eukaryotic cells that sort of helps RNA polymerase II to recognize promoters. Okay? All right. Well, talking about promoters, we see that promoters in eukaryotic cells are way more complex than they are in prokaryotic cells. In prokaryotic cells, we had a minus 10, we had a minus 35, and we really didn't have much more than that. We might have like the cap binding site or something, but there wasn't a lot of other sequences that affected transcription in prokaryotic cells. In eukaryotic cells, we might find 40 sequence elements that might affect transcription of a given gene. That's one gene. Okay? So there are many, many things. Why do we have so much more complexity for a eukaryotic cell? The answer is due to the fact that eukaryotic cells go through differentiation. All the cells in your body essentially have the same DNA, but not all the cells in your body have the same function. If I'm a bacterium, they all have the same DNA and they all have the same function. They all do the same thing. In our cells, we have skin cells, we have bone cells, we have intestinal cells, we have brain cells, we have you name it cells, but they've all got the same DNA. And so what that means is that some of those cells have some genes being made, other cells have different genes being made. Some of them have overlap, some of them don't overlap. So it's very complex for a given cell what pattern or what set of genes is being transcribed and translated, okay? So that means that we have to have signals, many signals for the cell to understand should this gene be being transcribed and translated or not, okay? Well, again, it's more complex than we're going to go into here, but what we see if we look at promoters in eukaryotic cells is we do see some things that are kind of like prokaryotic cells. We see a sequence called a Tata box. You'll notice I earlier called the Pribno box a Tata box. I frequently slip and do that. Okay? The Tata box is a name that's given to a sequence that's upstream, meaning ahead of, the transcriptional start site in eukaryotic cells. You'll see here it's labeled as a minus 25. Well, one of the differences in eukaryotic cells is it's not in a fixed place. It can be anywhere from about minus 30 up to about minus 100. We'll usually find a Tata box. You'll notice I said we usually find it. We don't always find a Tata box in front of all eukaryotic genes. We don't always find a Tata box. But any genes that we're making a lot of will always have a Tata box. All right? Any genes that we're transcribing a lot will always have a Tata box. But that doesn't mean that all genes have a Tata box. Okay? We see this thing here called upstream sequence element. And we may have many upstream sequence elements. Those upstream sequence elements, just like the Tata box, will have specific proteins that recognize those specific sequences and affect transcription. Some of them may be turning on transcription, and some of them may be turning off transcription, like we saw with the lac repressor. Okay? All right. Now, one of the ways in which cells, eukaryotic cells, know which genes to express is that they have sets of proteins that bind to sets of upstream elements. A skin cell may have one set of proteins, and all of those genes that have that upstream element will be transcribed in those skin cells. But if it's a bone cell and those proteins aren't present, those genes won't be made. Okay? So we can use the differential expression of the individual proteins as a way, that is the transcriptional binding proteins, as a way of determining whether transcription is going to occur. I should give you a term here I'm talking around. The term I'm talking around is a transcription factor. I want to define that for you. A transcription factor is a protein that binds to a specific sequence and affects transcription. A transcription factor is a protein that binds to a specific sequence and affects transcription. That can be positive, that can be negative, meaning it activates it or inactivates it. Okay. So the pattern of transcription factors that a given cell has will determine what genes what genes are turned on in that particular cell. Skin cells will have one set, bone cells will have another set, et cetera, et cetera. Some of them will overlap, some of them won't overlap. Okay. That's an important consideration. Okay. Well, one collection of factors that I'll talk about is something that you see here in this table. Okay? TF stands for transcription factor. And the Roman numeral 2 after the TF stands for RNA polymerase II. So TF2 something means it's a transcription factor that affects RNA polymerase II genes, in this case, messenger RNAs. Yeah, I hear the sigh. It's got a lot more to go to, yeah. All right. So what's happened? Then we have a letter after that. The letters are used to differentiate the different factors. Okay. TF2D, TF2D something else, TF2A, TF2B, TF2F, TF2E, TF2H, and guess what? These affect one sequence element. When I said there was a lot of complexity, I wasn't exaggerating. Okay? These proteins that you see here are involved in activating the Tata box. Okay? The Tata box. You see this thing here that says TF2D-TBP. TBP stands for Tata box binding protein. This guy right here, if there's one that you want to memorize in the table, that's the one. Because this guy is what's recognizing and binding to the Tata box. Then what in the world are all these other guys doing? Well, what we see with transcription factors is that there's usually a sort of a glomming on that happens. There may be one or two or even three that recognize a specific sequence, but the others recognize the proteins at that sequence. And some recognize the proteins on that sequence. It gets complex. We're not going to deal with that complexity. But suffice it to say that these guys here are all involved in starting transcription due to their binding of the Tata box. Yes? A promoter is not a protein. A promoter is a sequence in the DNA that's being recognized by proteins that affect transcription. Now, thanks for asking that, because it actually brings to mind something else that I didn't tell you. Another difference between prokaryotes and eukaryotes, RNA polymerase doesn't bind to the DNA. That's why we had this glomming on of proteins. These proteins that are glomming on are helping to tell the RNA polymerase this is where. The RNA polymerase is binding to these proteins, not to the DNA. It's not recognizing that promoter sequence. There are specialized proteins, in this case TF2D, that are recognizing the sequence and getting things opened up so that transcription can start. Yes? It doesn't mean anything specific. So D was, it was the fourth one they found. They found A, they found B, they found C, and then they found D. Yeah. Not a very glamorous answer. Okay, so those are the proteins that are involved in that. As I said, you don't need to memorize those, but I think TF2D you should definitely know. I show you this not to overwhelm you with information, which I could easily do. You guys have already seen that. Okay, so I try not to overwhelm you with information, but I'm just going to show you this just so that you get a feel for the complexity. There's nothing on here I'm going to require you to memorize. Okay, so relax and get a feel for this complexity. All right. What we see is what I just told you before. There's TF2D. TF2D recognizes that Tata box, and it binds to it. And it starts in motion a series of events that ultimately results in RNA polymerase II coming in and starting transcription. Okay? TF2D binds. TF2A and TF2B bind. RNA polymerase II binds. Okay? RNA polymerase II gets actually phosphorylated, and we see that happening right here. Let me back up here. We see RNA polymerase getting phosphorylated right here. And the phosphorylation, that is putting the phosphates on, that's what phosphorylation means, putting phosphates onto something. We're putting phosphates onto RNA polymerase II is necessary to start the polymerase doing its thing. We didn't see that in prokaryotic cells, but we see it in eukaryotic cells. Okay, well, all those other proteins that I showed you in that table bind, and they bind in a very specific order. We know the order in which they bind. One favors the next, favors the next, favors the next. There's a sequence of events, and as I said, the ultimate upshot of that is that transcription by RNA polymerase II starts as a result of that. It's very orchestrated. Part of the considerations in these various proteins being here and other proteins that I'm not telling you about are that we have to get access into the DNA when this DNA is all wrapped around all these other proteins. It's a very, very complex process. And we're only now beginning to understand how this unraveling and getting into these makes sense. If you're interested in this, come by and see me sometime, because some of these proteins that are involved in doing this are also implicated in longevity. Yeah. Very interesting story with respect to that. Okay. Another thing I want to talk about that we see in eukaryotes that we don't see in prokaryotes is something called an enhancer. We don't see enhancers in bacterial cells. Some books claim there's something like an enhancer in bacteria, but they're not the same as an enhancer in a eukaryotic cell. So let me tell you what enhancers are and what enhancers do. Okay? An enhancer is another kind of a sequence that affects transcription. What do I mean by another kind? Well, you'll see I'm not calling it a promoter because it really doesn't function like a promoter does. How is it different? Well, it's different by virtue of the fact that it is a specific sequence, but that specific sequence may be located thousands of nucleotides away from the transcriptional start site. That's one thing. It may be located ahead of the transcriptional start site, meaning 5' to it. It might be located downstream, 3' to it in the other direction, thousands of base pairs away. In some cases, it might be in the middle of the gene being transcribed. All right? And we know that this is the case because if we remove the enhancer sequence, transcription is drastically affected. Now, first let me tell you how this works, and then I'll tell you why we have these. Okay? How this works is depicted on here. An enhancer is a DNA sequence that also is bound by a transcription factor. Transcription factor says, oh, I'm going to go bind this sequence. Okay? The thing that binds it is a transcription factor. I call it an enhancer protein, but you can call it a transcription factor if you want. I don't care. Okay? The enhancer protein binds to that specific sequence, and what it favors is it favors the unwinding of DNA from the proteins that are there, the histones. So now it's favoring that loosening up of DNA so that all these other proteins can come in and get started. And what it does is what you see here is it loops around. That's how it can be located thousands of base pairs away. Once it has started to open this region up, it then interacts with the proteins like the ones I just showed you, the TF2D, the TF2As, et cetera, and because they can interact with the proteins at the enhancer sequence, transcription gets started. So the function of the enhancer in the enhancer proteins is to basically allow this region to get opened up so that transcription can get started. Now, why do we have these was the second thing I was going to say. And the reason that we have these is because they actually give us tissue specificity. Tissue specificity. When we talk about differentiation, and I said that we have certain proteins that will be present in one type of cell and not in another type of cell, and they can control the transcription, here is where we see these proteins. So if I open up the region of DNA so that transcription can occur in a skin cell, but a bone cell doesn't have that same protein, the bone cell won't have that region opened up. It won't be able to transcribe. But a skin cell will have that opened up and will be able to transcribe that. So enhancers give us what we call tissue specificity. That's something that bacterial cells don't have and, in fact, don't need because they all are the same type of cell. Question? Okay. He's speaking Greek again. I think that's what everybody's saying. He's speaking Greek. Okay. I'm going kind of fast, so I'll slow down. You're shaking your head. Yeah, he's going fast. Questions about this?
Can you have multiple regions for a single cell type?
Oh, good question. Can you have multiple regions like this for a single cell type? You certainly can.
So the cell, the part that opens up then, is it coding for a series of different things in the cell or just one specific protein?
Very good question. So the question here is, will this region open up basically correspond to one gene or more than one gene? In general, it's one gene, although it could be more than one, depending upon, again, how widely spaced these regions are. But in general, it's going to be for one gene. It's pretty amazing when you think about the fact that we have 30,000 genes in our genome. And any given cell is probably making at least 5,000 of them. So multiply the complexity of what I just told you by 5,000 for every single cell of your body, doing very specific things, because if they weren't doing the things specifically the way I told you, they wouldn't be the cell that they are. And it's a pretty remarkable thing. A pretty remarkable thing. Okay. Let's move forward. We'll get through this transcription. Okay. I'm just going to show you this very briefly because it's not a major point for transcription. But transcription factors bind to DNA. And when we look at proteins that bind to DNA, and we compare the structure of those proteins to each other, we see that they have some very, very common features. We call them structural motifs. Okay? Structural motifs, or in this case, binding domains. All right? But what they are common structural features. My hand has a structural feature that's like my other hand. They have fingers. When clenched in a fist, they're very similar to each other. Okay? So two proteins having common structural features, I can look at and say, oh, that looks like this one over here. That's what we're talking about. Okay? One of these is called a helix-turn helix. DNA binding proteins frequently have a helix-turn helix. And if you look at this, you can see how that works. Here, in the light blue, in the back, is a helix. That little purple line is the turn, and that's followed by a dark blue region that is another helix. Helix, turn, helix. And if you look at this, you can see that this helix, the light blue one, is interacting with DNA in one way, and this helix is interacting with DNA in another way. Okay? We can see that down here as well. Interacting one way, interacting another way. That helix, turn helix is a common structural feature. Another common structural feature we see are called zinc fingers. They're kind of cool. Okay? What we see is a sequence of amino acids in a protein. And if we look at this, we see that there's usually a pattern of cysteines and histidines, something like what you see on the screen. Those cysteines and histidines can bind to an atom of zinc and make a finger-like thing. We can think of, all right, here's in the middle of my knuckle is where the zinc is, cysteine, histidine, okay? And they're all interacting with that knuckle, and they're held together by that zinc that's in the point of my knuckle. That's what gives rise to this finger. This finger is very easy to think about because we can think of this finger as sticking its finger into the DNA. That's what it's doing. It sticks that finger into this DNA. And what it's doing, why does it stick its finger in there? Why does any part of a DNA or any part of these transcription factors, why do they form these structures? They form these structures because they have to read the sequence. And how do they read it? Well, in this case, the finger is literally taking the pulse of the DNA and reading that pattern of hydrogen bonds. And it says, oh, here's a GAA-GGGC. Because it knows by sticking its finger in there what it is. Yes? We see these in transcription factors, proteins that bind specific sequences in DNAs. Okay? That's where we see them. Other questions? Okay, the last of these, which I won't say much about, it's called a leucine zipper. All right? And it's just another structural motif. It turns out that these proteins, they first discovered them kind of an odd way, that they found certain proteins, every 7 amino acids, they found a leucine. They said, that's kind of odd. Why is it that every 7 amino acids in this protein, there's a leucine? Well, they started finding a bunch of proteins like this. Okay? Well, it turned out that when they looked at how the helix wound itself, every 7 amino acids meant that all those leucines were on the same side of the helix. And they could interact with things as a consequence of that. Okay. Enough of the structural motifs. The last thing I want to say about transcription relates also to eukaryotic cells. And by the way, you'll find these structures most commonly in eukaryotic cells of these proteins, because we have transcription factors are much more common in eukaryotic cells than they are in prokaryotic cells. Everybody want to stretch for a minute? Or should we plow through and get done? Plow through, okay. All right. I'm not opposed to stretching. All right, so RNAs get modified. DNA doesn't get modified, but RNAs get modified as a result of chemical modification. I mentioned earlier how the transfer RNAs are sometimes chemically modified. We see some unusual things happening inside of them. We don't fully know why. In eukaryotes, we see modifications happening to messenger RNAs that we don't see happening in prokaryotic cells. All right? One of these is something called a cap. Eukaryotic messenger RNAs have a structure that we refer to as a cap. I need to tell you what that cap is and why it's important, because it is. It'll be important when we talk about translation. If we look at the 5' end of eukaryotic messenger RNAs, we see something like what you see on the screen. And it turns out that what you see on the screen is a very unusual structure. It may not look like it to you, but trust me, it is. And I'll explain why it is. Okay? The unusual structure is it is a guanine-containing nucleotide. I know you looked at that and said, yep, that looks like a guanine, all right? All right? It's a guanine nucleotide that is attached. When the transcription started, by the way, the transcription started with this base right here. It didn't start with this one. It started with this one. So that meant that something got attached to the very first nucleotide after transcription occurred. This guanine nucleotide got attached, and it got attached in a very unusual way. It got attached as a 5' to 5' linkage. Not as a 5' to 3', but as a 5' to 5'. We don't see that bond occurring anywhere else in biology. Why do we care about that? We care about that. Maybe you don't care about that, but I care about that, okay? Very deeply I care about that, okay? I care about that because RNAs inside of cells have a limited lifetime. DNA doesn't. DNA is there forever. We want DNA to be there forever because it's the information that we need. But RNAs get made, they get used for a while, and then cells break them down. Why do cells break them down? They don't make the protein all the time. What if they need a protein for a certain period of time, but they don't need it anymore? Would you want to have the RNA floating around coding for more protein when the cell didn't need the protein? You don't. So what happens is RNAs have a fixed lifetime. They get broken down. That means that there are enzymes that will break down RNAs inside of the cell. Those enzymes are called RNAses, R-N-A-S-E. An RNase will usually start chewing, that is, breaking down an RNA starting at the end, and it moves in. It starts at the end, and it just starts gobbling up nucleotides one at a time, chewing them up. Well, it turns out that the worst place that you can break down a messenger RNA is at the 5' end, because that's where the start of the protein coding is. We'll see that cells do this starting at the 3' end. So the 5' end, because it has this structure, partly to protect it from being degraded by RNases. It's there to protect the cell from being degraded by RNases. So the 5' end is covered, it's protected. The RNA is going to be degraded from the 3' end. Okay? Second function of this cap is we will see that it's involved in the process of translation as well. Okay. Another thing that we see in eukaryotes that we don't see in prokaryotes with respect to messenger RNAs is at the other end of the messenger RNA. At the 3' end, we see a sequence that we never see in prokaryotic cells. And the sequence is interesting and odd. The sequence is the following. Here's the entire messenger RNA, and then we reach the end of where the RNA polymerase stopped, and we see the following. We'll see 200 or 300 A's put on the end of that messenger RNA. Kind of odd. Well, not kind of odd based on what I just told you about the RNases. RNases start at the 3' end and they start moving inwards. Remember the telomere? What was the function of the telomere? It provided protection, a buffer as it were, so that each time we lost a little bit of sequence, we didn't lose anything important. Those A's are there to lengthen the amount of time the messenger RNA is floating around. The RNase gets on, it chews off a few, and it falls off. It gets on, it chews on a few, and it falls off. The longer the poly-A's, the longer the amount of time the messenger RNA will be around. The shorter the poly-A's, the shorter the amount of time they will be around. Okay? As we will see, poly-A's also have a function in translation. So both the 5' end and the 3' end both have functions in translation. All right. I've already shown you this one, so I'm not going to drag you through it. Okay? I told you about splicing already. Splicing puts together regions of a messenger RNA that didn't start together. Okay? The things that get put together are called exons. The things that get removed are called introns. Okay? We don't see this in prokaryotes. We do see this in eukaryotes. We just finished transcription. Yay. You guys are going, man. All right, questions or comments? You changed your mind on stretching? Okay. All right. Let's talk about, and oh, by the way, you guys wanted to have extra credit. Maybe this would be a good place to do some extra credit. What do you think? Okay. We'll do the extra credit, but we're not leaving because I wanted to do some things after that. I'll give you 5 minutes to do the following. Take out a piece of paper. On the paper, please write your name and your ID number. Your name and your ID number and a joke. The better the joke, the higher the grade. I'm joking, but a joke would be good, right? I don't care how bad the joke is, just something that you can write. Well, if you can do it in 5 minutes, yeah. If I hear any really good ones, I will pass them on to the class. Make one up. Knock, knock, who's there? And yes, you may use smartphones to find a joke. I see some of you doing that. Nacho cheese? Is that the one? Everybody got it? Let's take about 1 more minute. Everybody got it? Okay. Hopefully that livened things up. So at the end, I'll ask you to come up and bring it, hand it to me personally. That makes sure way I make sure people don't just leave and have the extra credits here. I can go home, right? So hand it to me personally when you're done at the end of the period. Okay. Let's talk about translation. This is the last of the central dogma. The central dogma, you remember, was DNA replication, transcription, and translation. We've done replication, we've done transcription, now we're going to do translation. Translation is the synthesis of protein using the genetic code that's in the messenger RNA. And since the messenger RNA was copied from the DNA was the ultimate source of that information. Okay? There are three main steps to translation, just like there are in transcription: initiation, elongation, and termination. And we'll talk about those processes. In addition, there's one other step we sometimes put onto translation. And that is a step that involves what we call activation. All right? This involves the attachment of the amino acid to the transfer RNA. And I'll say more about that as I get going further along. Okay? So if we think about this, that has to happen in order for translation to occur. All right. Well, I bandy about the genetic code. We all hear about the genetic code. And the genetic code, I'll remind you, is a sequence of 3 bases that codes for or specifies one amino acid. There are 3 bases that make that up, and each of those bases can be any of 4 different bases. So there's a total of a possible 64 different 3-base sequences. The 3-base sequence is called a codon. A codon is found in a messenger RNA. Remember that. A codon is found in a messenger RNA. A 3-base sequence that is complementary to the codon is called an anticodon. And it's found in the transfer RNA. Codon in the messenger RNA, anticodon in the transfer RNA. What you see when somebody shows you the genetic code is they're reading you the sequence of the codons. Okay? Well, how do I read this? It turns out it's actually fairly simple. We see U is the first at the 5' end. The middle one here is a U. And if I go over here to this one, let's say this A, the sequence would be U, U, A, and I can read U, U, A, and see that would correspond to a leucine. I can do this for any of those 3-letter codes. As I look at the pattern of the genetic code, I see something interesting. I see, look at this, CUU is leucine, CUC is leucine, CUA is leucine, and CUG is leucine. It means that I could say that CUX, where X is any base, could be leucine. And no, you're not going to memorize the genetic code. Okay? But it's not just leucine that has that property. If I look over here, here's CCU, CCC, CCA, CCG, and all of those make proline. This code has what we call redundancy, meaning more than one codon can specify an amino acid. Why do we have redundancy? Well, there's only 20 amino acids. There are 64 possible codons. You can do the math. That means you're going to have more than one codon for many amino acids. Not all. There's a couple that only have one amino acid, or one codon for an amino acid. A really good example is methionine. Where's methionine? Right there. AUG is the only codon that specifies methionine. Tryptophan's another one. Here's UGG, the only one that specifies tryptophan. If you look at this carefully, you'll see there's also 3 codons that have the word stop beside them. UGA, UAG, and UAA. They're called stop codons because when the ribosome encounters them during the process of translation, it knows this is the end of the code. It stops translating at that point. Just like we have a stop codon, so too do we also have a start codon. AUG, methionine, is the start codon. Essentially, every protein that's made on the face of the earth starts with a methionine. And by the way, the genetic code that I'm showing you is the same for you as it is for the bacteria that are in your gut. It's the same that a virus has. There are only very, very rare exceptions to the genetic code, and those are very minor, like maybe one codon being different, across all of biology. The genetic code is what we call universal. This universality of the genetic code turns out to be very useful. Let's imagine that I am a biotechnologist, and I decide I want to have a bacterium make human growth hormone because human growth hormone is needed, and it's very, very expensive to isolate. People used to have to isolate it from corpses. That was the only way they could get it, was they'd take the blood of corpses, and they would extract human growth hormone from that. If I get the gene for the human growth hormone and I put it into a bacterium, I can make that bacterium synthesize human growth hormone because the code is the same. That's really useful. Very, very useful. So the universality of the code, all right, allows us to do a lot of things. Okay. That third position that I just described to you isn't as important as the first two because in many cases it doesn't even matter. The third position is something we call the wobble base. Because for many of the codons, the third position, it doesn't matter. I could have CCX, and I've always got proline. Okay? Here is the wobble position. If we go 5' to 3', it reads like this. Here's the anticodon that's complementary to it. It's going 5' to 3' in the other direction, and look at what we see. At the 5' end of the anticodon, which corresponds to the 3' end of the codon, I've got a base I haven't seen before. That base is an I. It's called inosine. I-N-O-S-I-N-E. Why is there an I there? Right? Well, it turns out cells aren't stupid. Cells are extraordinarily efficient. I can pair with 3 of the bases over here, not just 1. It means that the cell doesn't have to make a separate transfer RNA for every possible codon. It can make one that'll work for at least 3 of the 4 for a given codon. That makes it simple. Okay? So instead of needing 64 transfer RNAs, the cell gets by with about 20 or 30. Simplicity is good. And it happens because that third position is not as important. The reason we don't have I in DNA is because I can pair with multiple things. And we don't want it pairing with multiple things in DNA because that would give rise to mutation. We don't care about that here. This has already been synthesized. Okay, so there's I. I can pair with A or C, or no, you don't need to memorize that. I'm just showing you that, okay? It can pair with any of those. It turns out that G is capable of pairing with U. G can pair with U. Okay? That happens in RNA. G can't pair with T in DNA, but G can pair with U in RNA. All right, you guys have been very good and patient today. I've got a song to finish everything off, and then we'll call it a day, okay? The song is called The Ribosome. Oh, beautiful with RNA to make the peptide bonds. You hold tRNA so it can pair up with codons. The ribosome translates